My name is Sydney Brenner and I'm a geneticist. I would like to call myself that. I was born and educated in South Africa, and I spent 40 years or more working in Cambridge in the MRC laboratory of molecular biology.
I'm Barbara Meyer. I'm an investigator of the Hughes Medical Institute, and I'm a professor of genetics at UC Berkeley. One of the most provocative things you said to me once, when I was your postdoc, was there's an incredible importance to be ignorant in science. And I thought perhaps your own beginning in science reflected what that statement meant. And I wondered if you would tell us a bit about your early life in South Africa and how you managed to train yourself to be a scientist, because you did it yourself.
Well, I grew up in South Africa, very small town. South Africa was provincial by all measures, and I had a very early great interest in the natural world. And of course, when one goes to school, one doesn't hear too much about this, but I made the great discovery of a library, the public library, and that's where I went to get all my information. In fact, it was my demands were insatiable, that at the age of, I think it was 10, I was allowed to join the adult library, which of course had more books on science there. And so the habit of being self-taught has stayed with me all my life.
You must have done your own experiments as a child.
My own experiments I got a book which I still have my copy of, called The Young Chemist. Written by Sherwood Taylor, which told you how to, make explosives and all the sort of this. And by the time I was 10, I already had great experience in what I called garage chemistry. But I was more interested in biology than chemistry, and one of the things I discovered was I used to make extracts of plants. And I discovered, to my amazement, that one of my extracts, if I put vinegar in it, it changed color. And so, later I learned that it had already been done. It was called litmus paper. But I found that a very great interest. And when I went to university, only because I could get a bursary from my local town council to study medicine, which I started studying. I was 15 when I started medical school. I had to do botany, zoology, chemistry, and physics in the first year. This was the Scottish system. And one of the things that I will remember, there was a professor in the botany department who worked on chromatography, and after hours I used to go and work in his lab. I'd never seen chromatography, and all this wonderful separations of plant pigments. And I've always been fascinated by colored things, so much so that much later I learned how to synthesize my own dyes and did lots of things on staining. And then it was discovered that I'd be too young to qualify to practice medicine. You had to be at least 21 to do this, and I would graduate before this. So I took a year off to do a medical BSc, which was a real eye-opener. And there we were, 11 or 12 of us in one room, learning how to do experiments, doing all sorts of things. And we did everything from paleontology to studying the diffusion of dyes in agar. I did an honors degree, And this is where I got interested in genetics.
But before that, if I've calculated this right, at the age of 18, you published your first paper in Nature, and it was on porphyrin fluorescence.
It was in the South African Journal of Medical Sciences. But I went on with my own work because I decided I would want to do genetics. Effectively, it was still not genetics of breeding, but cytogenetics. And so I read a book. It was C.D. Darlington's The Handling of Chromosomes. And so that's when I learned how to make acetic orcine squashes. I spent a lot of time with onion roots. I even made preparations of salivary bands by getting hold of some fly larvae and doing this, because it was still concentrating on the microscope that I thought was the important thing. However, I then took a third year out and I did a master's degree. It was by thesis. And my thesis was to count the chromosomes in the elephant shrew. And I wrote a lot of papers. I actually got it wrong, I'm unhappy to say. I got half the number. So I must have mistaken what I thought was a mitotic cell, but it was a meiotic cell. So, but in any way, nobody else was doing this in the entire country.
Or the world.
I supported myself during this time by being a technician in the Department of Histology. Just to complete this, I then at that stage, Legros Clark, professor of anatomy, came out to South Africa and offered to me that I could go to Oxford to work in his department. I then asked a lot of people and they said, no, don't do it. There's no future in doing pure science because you'll have to get a job in a medical school and you'll need a medical degree. So very reluctantly, I went to complete the last 4 years of medicine. But I moonlighted by doing research in the lab at the same time, and I continued to publish papers all through that time.
And mostly by yourself?
Mostly by myself. One paper I was very proud of, which involved, the concept was, why don't I centrifuge the components of a liver cell and see if the cytological elements line up in the liver cell the same way they line up in a test tube. Now, in order to do this, I built an ultracentrifuge. This was a little air turbine which had been designed by Beams and King and which was powered by compressed air. And I had the problem of measuring the G in this. And I did it, of course, the first time I did it, I took a little lead weight and tied it with a piece of wire to the center of the centrifuge and accelerated the centrifuge until the wire broke. I then knew the weight, the size of the weight. I then took the same wire, put a bucket of water and filled it with water till the wire broke. And then, of course, just a matter of what you knew the weight at 1 G was this. However, later I got more accurate measurements by measuring the speed stroboscopically.
So when you finally did go to Oxford, your intention at that point was to think about phage resistance, is that right?
My intention was to study molecular biology, but the subject didn't exist. And so I wanted to learn more about physics and chemistry in relation to biology. I first applied to the biochemistry department in Cambridge, but they never even replied. So then the president of my university was a man who came originally from Oxford, and he said, why don't you go and work in Oxford with Sir Cyril Hinshelwood. And he had written a very interesting book in the 30s called The Chemical Kinetics of the Bacterial Cell. So that is how I got to Oxford. And of course, Hinshelwood was very notorious at the time because he didn't believe in mutations. He thought all things were adaptive. And what he wanted me to prove, that phage resistance, like drug resistance, could be adaptive. And of course I showed that it wasn't. That's when I started to work with phage. In those days, when you did, I was given a pressure cooker, 24 glass Petri dishes, each of which weighed about 1 kilogram. The glass was so thick, and a certain number of pipettes. We had to wash up everything and sterilize everything ourselves. So it was quite tricky to do experiments, just the logistics of it. In fact, I swore them that day that if ever I'd have a lab, I would saturate it with provisions. I repeated the Luria-Delbruck experiments. I must have been the only person in the world to have done it, and repeated them because Hinshelwood, he realized that these are quite important because you're counting clones, you see, not counting the sum of additional events. And so I repeated them, and this gave him doubts about it. And then finally I did a very interesting thing. I mean, it's a paper he submitted for me. I did an absorption isotherm. I took normal E. coli and I added phage till it was saturated, you see? And I measured this by how much phage did it remove from the solution. And then I did one with a resistant mutant, showed it didn't absorb anything. That impressed him as well. And I remember crystallizing this explanation to him by telling him the difference between chastity and impotence, which I've used very often in my life. You know, the difference between can't and won't. And of course, that's a very fundamental thing, both in science and in life. Well, during my time in Oxford, the people I met very early is a man called Jack Dunitz, who's a famous crystallographer in Zurich. And Jack had been at Caltech, and he knew all the people in the DNA. And he said, well, there's these people, you know, called Watson and Crick, and they're working on the structure of DNA and so on. And then he said they had a structure. And so Jack Dunitz and I and Leslie Orgel, on a morning in April 1953, went over to Cambridge to see the structure. I've written a lot about this. To me, as I've said over, it was a revelation. See, I'd entertained all kinds of ideas about how you might determine the structure of DNA, how DNA might guide the synthesis of proteins. They were all hopelessly wrong, you see. But here, this just came through, here is the truth. We have to follow it.
By seeing the structure, you realized how replication
have to
would
follow
occur.
it. And I then realized, you know, I mean, it became clear what the coding problem was going to be about. And that's how I really started to do things seriously in molecular biology.
But did you have the opportunity at that point
Yeah,
to start
I did.
those?
What I did know more about was I knew more about protein chemistry than either Francis or Jim at that time. And I realized, and so I started to work just on sequences, protein sequences. By the summer of 54, when, I have to tell you that Demirats came to visit Hinshelwood. And of course, Demirats was the famous geneticist who was the director of Cold Spring Harbor. And Hinshelwood introduced him to me, and I began to talk, and it was very funny because for the first time he realized he has someone who knows something about mutations and chromosomes. He went and he closed the door. And then he said, why don't I come to Cold Spring Harbor? And Demirats arranged for me to get a Carnegie Corporation fellowship, which took me to America for 4 months. And I then went to Cold Spring Harbor, spent the summer there, went up to Woods Hole where Jim and Francis were. And that is where there were lots of discussions about the genetic code. Gamow had got interested in coding, and he'd written a paper to Francis and Jim saying that, which essentially was the overlapping triplet code, but which was called Gamow's diamond code, because he saw there were cavities on the DNA which he called diamonds, and that his idea was the diamonds could be made to code for the amino acids. The striking thing is that there are 64 triplets, but if you take Gamow's degeneracy, you'll end up with 20 amino acids. Magic, magic, magic. And so, but it was wrong. Gamow was in Woods Hole. And what I had by that summer was a Poisson distribution of dipeptides. And the question was, if Gamow's code was right, would there could only be 256 dipeptides, because that was the nature of the code. But if it was wrong, there could be 400. That is, all sequences were possible. There weren't enough to disprove the thing. And so I did a Poisson distribution, being a phage man, that's the only mathematics I knew. And I then showed that it fitted better to 400 than a 256. But I showed it to Gamow, and he and Alex Rich kind of did the same thing, but mentioned that they had seen this in a footnote in a review they wrote. In the meantime, I realized you didn't have to have complete information. And so I wrote a paper entitled On the Impossibility of All Overlapping Triplet Codes, which we circulated initially to just the 20 people who were members of the Thai club. And Gamow then said, you've got to publish this, and he put it into PNAS for me.
Can you give me more information about
Gamow formed a club called the RNA Thai Club, and they were mostly Gamow's friends. There were 4 bases that were honorary members, and Wendell Stanley was one of the honorary members. But the other 20 members, each corresponding to an amino acid.
Who are you?
I was right at the end. They were in alphabetical order, and Francis was tryptophan, Leslie Auger was tyrosine, and I was valine, just got in. And I still have letters from Gamow saying, "Dear valine," you see. Gamow was an amazing character. The other members were, Jim was a member, Jim Watson was a member. A lot of them were friends of Gamow from Los Alamos, where he was a consultant on the physics and weapons programs. This RNA Thai Club circulated a lot of papers. I think Francis and I were responsible for most of them. Francis wrote a brilliant paper, which I think he circulated in the beginning or the end of '54, on the adapter hypothesis, which was the whole idea that nucleic acids were organized for information, proteins organized for catalysis. So this meant that we had to have some bridge between this. And so what he said is that we would have an adapter, which is a word I invented for that at the time, and that adapter would have, there'd be 20 adapters, 20 amino acids and 20 enzymes to put the amino acids onto the adapter. Today we all know this is true, it's called tRNA. But in that time, all the biochemists said this is impossible, because if the enzymes existed, we would have already found them, and since we haven't, they can't exist, you see. So, well, I think the important thing of Gamow is that he drew attention to the nature of the problem. Everything was contaminated by this idea that the spacing in the polypeptide chain was about the same as the spacing between the bases. But informationally, that would only mean you could have 4 amino acids. I remember there was what we call the naive biochemist's code, which was that you did it with all the doublets, and the doublets were 16, and then you also used the single bases. So 16 plus 4 is 20. You'll realize that getting the magic number 20 was really part of the game. And of course, this is the fate of that most beautiful code called the Comolus code, in which by just a few very simple arguments, you can take 64. And if you ask, if the code is only read in one phase, then it means that overlap phases, if it's read 3 at a time, which we all assumed, overlap phases must then not correspond to bases. And if you take each of the triplets and cyclic permutations of them, there are 3 cyclic permutations of each of these, only one of which can be selected. Right? And that gives you 20. And the amazing thing is, there's the magic, the number. And of course, it's very elegant, but it's untrue. See, that's when you began to realize that mathematics may be the art of the perfect, but biology is only the art of the satisfactory. If it works, it's going to be good enough. Okay, so that's something which I think still is unclear to most people today. Anyway, this became then that we would have to settle the nature of the code by experiment.
These are my favorite experiments of all time, so I really want to hear you talk about them.
So the idea was, why don't we try to find we'll first do a simple experiment. We'll prove what we call the collinearity hypothesis. If there's this correspondence, then the order of the mutations in the gene should be the same as the order of the changes in the protein. Now, one thing we did know is that we had a tool to measure chemical changes in the proteins. And of course, the most important thing is that what was possible, that Vernon Ingram had discovered, single amino acid changes in proteins could be detected by fragmenting the protein. So we knew that most mutations change a single amino acid, not more than one. However, we decided to embark on this, and it's a long odyssey in which first you had to find a gene, then you had to find a protein. And the gene you had to be able to do fine structure genetics, which of course Seymour had done with the R2.
And
That
excuse
was
me,
when
when did you first meet Seymour? How did you
Seymour I
find
met
Seymour?
at Cold Spring Harbor in 1954. He was there for the summer with his family. And as I've said, there was Seymour going around with 5 mutants mapped internally, the gene. There was I going around carrying a lot of amino acid sequences because I was working on trying to crack the coding problem. And if you put 2 and 2 together, you can now make the whole project. So this became the kind of problem to solve. We chose collinearity because by genetics we at least knew we could do, and especially by fine structured genetics, we could at least map the mutants in an order. So we said, well, that was the great thing to do. And so, of course, the perfect gene for this purpose was the R2 gene of Seymour's, but how would you get the R2 protein? And we took another thing because when I had to go back to South Africa in the end of '54 for 2 years to serve out my bond, I then started to work on something that I knew had to be a property of the phage coat called cofactor dependence in tryptophan. And I thought, well, it's not beyond the wit of a person, we'll go and find these more mutants. And so I knew here was something, and I could actually show recombinance between them. Being phage, you could do this because I could select for ones that were independent. When I returned to Cambridge at the beginning of '56, joined the unit, Francis was able to get me a 3-year position. I came from South Africa with a family for a 3-year position to do these experiments. And this was in the unit, was called the Unit for Molecular Biology. And we started to set up biology in this. We started in a marvelous office down in the basement of the Cavendish, which had 7 desks. And in fact, all our washing up was done in that same room, you see. And then we got another lab. And we also got a hut there outside the Cavendish. But the other lab was really tremendously important. It was in a building which has since been demolished called Old Anatomy. Old Anatomy is where the French tried to build an atom bomb during the war. And I only realized this when one day I put a Geiger counter on, and it's just amazing. You know, the walls were full of all sorts of things. We had to come and dig them away. However, we had a kitchen outside, and we had 4 benches in this. It was like there was a glass sort of conservatory where we put our kitchen. And that's where we started to do phage work. And that is where most of the work was subsequently carried out. We had a great time because when you don't have space, and we didn't have any space, we began to invade. At one stage, we were in 7 buildings on the Cavendish site. And we noticed that there was a long building which housed a whale. And the whale had sort of got a bit moldy around the edges, and so they removed it. And Francis went to the professor of zoology and asked whether we could borrow this room. And it was about 50 feet long, but it was very narrow. And so what you could do is you could have one bench all the way along the windows, and so it was like the Mad Hatter's tea party. You get everything there, you do your experiment, then you move on to the next place while someone comes up and cleans up the mess. But it was hilarious and that's where a lot of these experiments were done. But before that, still working in sort of corners of the Cavendish, Seymour and I did with Leslie Barnett, began to study proflavin-induced mutants, you see. on the grounds Seymour got interested in chemical mutagenesis, so was I. One wondered whether one could not only measure the position of changes on the DNA, but measure what bases there were. And that said, boy, you know, we could, if you like, sequence the DNA by genetics. Okay? And this became the great sort of period where we were going to solve the code by sequencing the DNA by genetics. And it only, I'll say in a moment how it happened that it only got matured later. So one of the problems was the following, that whereas all the base analog mutants that you could find, they formed a coherent map. There was hardly any overlap between this and the spontaneous mutants. And so whatever you assumed about the induced mutants, the fact you didn't seem to see them with spontaneous mutants said that these were different. And so base analogs, it was believed, converted one base by transition mutants, converted Gs into As, which is the same as saying Cs into Ts. And so it was hypothesized that the others would be mutants that perhaps were transversions. Right? And that was one of the things. But I remember that Francis and I went to have lunch in the Eagle pub. And we came, I said, this is quite interesting, because I said, you know, there could be additions and deletions, which might explain why we never found any, you know, why they were always seemed to have very stringent phenotypes. In fact, I said, I think I can prove that, because if that is the case, if we look for mutants in a structural protein of the phage where we must have it, we will never find a proflavin mutant, we'll only find base analogs. And I went actually back to the lab to do that experiment. That's the beauty of those days. You could have an idea and go back to the lab and sort of have results over the weekend. And in fact, we wrote a paper on this, which just said that these have a different kind of property, and they could be thought of as having additions and deletions. Now, at the same time, Francis got into the lab himself.
Sydney, was it unusual for Francis to be doing experiments?
It was very unusual for Francis to do experiments. He went into the lab to try and purify tRNAs to prove the adapter hypothesis. Actually, it was only years later that one was able to do this. But he did try, and of course it was very primitive. And then he went into the lab to work with the phage. And that was actually tremendous, because at that stage we had this wonderful room. And so he decided he would work on these mutants. Okay? And he would test one of the hypotheses he wanted to test is that the DNA folded back on itself, okay, and the mutants were made so that every mutant had a symmetrical counterpart. And that if that were the case, which of course is actually true for tRNA, amazingly, we could suppress one mutant in one place by a mutant in another place. And so he started to look for suppressors. And at first it was very messy, but then what he discovered, they could get distance suppressors with these acridine, the proflavin-induced mutants. And there was this astonishing result that we could take a mutant and suppress it by one further away. And so we suppose one is an addition of one which we call plus, then it could be compensated for by a removal of a nucleotide further away, provided the protein was permissive. We worked in the first part of the R2B gene because Seymour had shown that that part could be dispensed with by the fusion deletion. And Francis then had this whole thing of which we called uncles and aunts and nieces and nephews, and you could put them into a pattern. And the question arose, how could we prove from this pattern that, actually how many of these, if you found you needed 3 pluses or 3 minuses, then we'd know the code was triplet. If you needed 4, it was something else. So we decided we could prove that the phase was modulo n by simply finding how many of these you could put together, right? And of course, this is quite a tricky thing to do because everything is mutant. But I said to Francis, no, the way we must do this is, suppose we have 3 mutants, A, B, and C. Then we will make the AB doubles. Right. We'll make the BC doubles. Both of these are mutant, okay? They can't give you a standard wild type by recombination because they share B. And that means if we get wild types, it must be a triplet code. So we set out to make all these doubles. At one stage, Francis very quickly realized that we had to be careful of what he called barriers. The barriers turned out to be nonsense mutants, that is, chain terminating mutants in the different phase. And so that is the nature of the experiment, which is, it's an interesting experiment because certain things had to be, certain results had to be excluded from this. And we wrote this paper on the triplet nature of the genetic code, proved entirely by genetics. Nothing else. Proved entirely, as I used to tell people, by just looking at holes on a petri dish. Not even looking at single organism, but looking at a colony of organisms. And it was marvelous because, see, what was that? You could look at something in this macroscopic world that is trivial and make deductions about, the intimate molecular architecture of the gene. And there's been nothing like that since.
When I read the early papers, I was struck by how thorough you were in tracking down all the exceptions and what that meant to you. And I find that students these days don't do that and don't even believe it's important. And yet your work is a testament to that importance. And I would like you to speak about that.
We had hundreds of anomalies, which we didn't tell anybody at that time. And when we later finished all the experiments, we wrote a big thing called a telephone directory, full Trans Royal Society, of every R2 mutant that was there, with every anomaly resolved. And the interesting thing,
I hope.
In the appendix, in the last part, and what is interesting is that every anomaly had a different explanation. Today I find that there is no student that knows how to interpret a negative experiment. And very few know how to interpret a positive experiment, but no student knows how to interpret a negative experiment. And what they simply do is just buy another kit and try again. And the negative experiment is the one that really teaches you, especially if you believe, there's a good reason for it being negative. So I think the analysis of experimental and work today is, if it doesn't work, then we try it again with this. That I think is now missing. There's a few more experiments
Oh,
of the same
course,
ilk
yeah.
that we were able to do. For example, we actually decoded the nonsense mutants
tell us more about
by this, by actually
really
this
clever.
business of mutagenesis, you see. So let me just see if I can recall. Okay, now this depended on the following. So we started with a mutagen called hydroxylamine. Okay, now what we knew is that hydroxylamine changes C into U, or a U equivalent. So now consider that you take a phage, again it's our friend the R2 gene, and let us suppose that the chain that is copied, okay, actually contains the mutant C. That is copied, in fact, into mRNA. If that were the case, then we would get two kinds of mutants. Those in which the modification in the parent phage was already mutant, that is, would be an R2 mutant, okay, and those, in fact, where it wouldn't be a mutant, it would come through, but be a mutation once it had replicated,
Yeah.
all right? And so we sat down to do these experiments by taking the strains through K first. We would filter all the ones out in the sense strand. By taking the ones in B, we would get both. And we would just find out which were the mutants so that we could tell from this experiment. And it was an enormous experiment because we had to collect an enormous amount of mutants to find the amber mutants and to find spectra of the amber mutants. So every day we sat down and picked and stabbed, you know, hundreds of things. We did this every day till we collected several hundred amber mutants from the two conditions and compared the spectra, okay? And we could then tell that this one had a C in the first, you know, in the position, whereas this one had a G. And by those arguments, this is the only case
Where you could sequence
where
the
we
DNA.
could actually sequence a triplet, right, by this. Now, we did a lot of other work. We actually, although all the experiments we did, we focused on the head protein of the phage because there's lots of it. And Fred Sanger was at that time trying to develop radioactive methods of doing protein sequencing. And I said we could do this with phage because we don't have to purify anything. The phage is 70, 80% of what's synthesized. And this then allowed us to do some very interesting experiments. So first of all, we could fingerprint the heads, and we could fingerprint them, the distribution of amino acids in it, by looking at peptides, if we label them with histidine, because that's how Fred was going to do it, by composition. And this is when Tony Stretton and Anand Sarabhai joined this work. Now, Anand was a student of mine, and we used to talk about the luck of the Sarabhai. And he did two experiments. This one, he decided, we had a long discussion one day on how many kinds of nonsense are there. See? So first of all, the idea is you could have a nonsense, although we didn't know what happened, if you didn't put if you left out a tRNA, or if you had a mad configuration that did it. Of course, we somehow had to explain how things terminated. So the idea became that certain classes of mutants would terminate, right? Since that is something we could tell, Anand was the first person to check out that he got a fragment of the head protein. Francis said he didn't think it would work. Although Francis and I were very close collaborators and friends, we wrote very few papers together. I think there's only about 2 or 3 that have both our names on.
Why was that?
Well, one of the things that we were very strict about, we only put names of people on papers that actually did the work or made a significant contribution.
Times have changed.
Times have changed, you see, because you had to have had a key idea. And I remember Francis saying, a key idea is not, have you tried a different concentration of magnesium, you see. So I think that it recognized the people that actually did the work. And so we didn't have what I call the caboose theory of scientific publication, where you get everybody in a long line and the head of the lab comes last. In fact, I think now it's people go from being the first author of a paper to the last author of a paper without ever reaching the age of accomplishment. So I think and this was very good because we discuss all these things, we'd have these endless discussions. The discussions on nonsense went on for days, you know, in which we tried to classify, you know, there was nonsense called gibberish. That is, it was nonsense as far as the protein was concerned. There was nonsense which was a whole. And so that was a very important thing. But I've left something out because while all this is going on, we had a visit in April 1960 by Francois. He came to Cambridge and we were all talking about these mutants, the operator mutants. And in fact, it was extremely interesting to ask what actually made proteins. Because if you look at 1958, there are papers by myself and Francis in a Brookhaven symposium, which we've come to the conclusion that there's this terrible paradox about the composition of RNA not corresponding to the composition of the protein. And it actually was, the arguments about this were very similar to the arguments about junk today, right? So what we said was the following. Suppose when you measure the DNA, the DNA is composed of 2 fractions, okay? One of which corresponds to the structure of the ribosomes, which was supposed then to contain the code, because the code would have to be uniform in some sense, and then the other consisted of junk. Right? That was exploded when Suoka and Doty showed that the composition was very fine-grained and wasn't distributed in lumps. So we now had this problem of how do we explain that an organism with a very high GC makes the same as proteins, although the composition of the ribosomal RNA is the same. Now, there were, of course, lots of theories about this. In fact, they entertained in Paris even the idea that DNA might make proteins directly.
Amino acids would line up on the DNA.
The amino acids would line up on the DNA. The question is, how would one prove what was the information intermediate?
But what was your own personal insight?
No, the insight was the following. It was this base composition paradox. And I remembered there was an RNA with the same apparent base composition
this is a
as the
transient
DNA with the T4 phages, as had been discovered by Volkin and Astrakhan a long time ago. Yeah. There was this RNA. It was actually thought to be a precursor of DNA. And I suddenly realized that, since I knew that T4 shut off all host protein synthesis, that if there was this so-called messenger, it would be that RNA was a candidate because it was the copy. It copied the base composition. DNA, it was just fortunate that T4 is much higher AT than E. coli.
So where were you when you
We were all in a room. My room's in King's College. There were a number of people sitting there. Alan Garron was there, Francois Jacob, Francis. And I leapt up and I said, Volkin Astracana. And Francis picked it up immediately. Yeah. But most of them didn't know what we were talking about. And I know that I went home with Francois and said, this is the way we're going to prove it. All right? Now, it turned out, this is April, turned out Francois had been invited to Caltech to give some lectures in the summer. And I went there at the same time. And we had 3 weeks in the summer of 1960 to do this experiment. We were going to do it by showing that new RNA goes on to old ribosomes. How did we know they were old? We would do density. We'd do density labels. Who was the guy that had all the materials? Matt Messelson. Yep. Okay. So Francois and I turned up there to do this experiment. And we did several experiments. We failed. The ribosomes came apart. Matt Messelson had more pressing things to do on the East Coast. And we just got really depressed. And there's a story which is recounted by Francois in his autobiography. We went out to the beach, and I suddenly realized that, you know, we put in thousandth molar magnesium, but I suddenly realized we're doing the centrifugation in 8 molar cesium chloride. So I leapt up, shouting, it's the magnesium, it's the magnesium. And we went back, we just had time to do the last experiment, where we just put a hell of a lot of magnesium in. And I will never forget this, because it was the last chance experiment. And during the equilibrium centrifugation, our centrifuge broke, the one we were using. And Renato Dulbecco said, we could use the one, his one. And so I carried this tube, I mean, with the sweat pouring off me nervously, saying, don't drop it, because what Francois did in the course of this experiment the day before is he missed the tube with a P32 and it went into the water bath, which we hid behind the Coca-Cola machine and pretended not to know anything about. Every time I visited Caltech, I would look behind the machine, because I waited a couple of weeks and then told them about it. Of course, the P32 had decayed, but I carried this like, you know, like it was the crown or something. And I took it to Renato's, and it then had to run for another day. These centrifugations went on for 2 days. And I then had to make the collections. And, you know, which we did by piercing the bottom of these little tubes with a needle, and then collecting the drops. And once I said, I'm not going to look, you see. And I was very nervous, but I did do it. We collected these hundreds of drops, okay? We then had to put each of these drops on a planchette and put it through the nuclear Chicago counter. Now, that night was the night that John Kennedy, Kennedy became the Democratic nominee for president at a big thing in Los Angeles, okay? And everybody was gathered around looking at this, and we could hear the sound going. And we sat down to look at our counter. And what we saw very satisfactorily, that we began with nothing. Usually the other ones had already, the RNA had come down to the bottom of the tube. There was a smear, but there was no smear. And then it started to go up. And I remember that as they were cheering there, we were cheering. And it went right up, and then we said, it's enough, go down, go down, go down. And the curve went down, and it was perfect. I mean, the oldest RNA, which was new RNA, was on old ribosomes, you see. And we could then show there were no new ribosomes synthesized. Then, of course, I then went back to England. In the meantime, all these other phage experiments were going on. And then we had to do all the controls. And of course, those took quite a bit of time. But by the end of the year, we had been able to write a paper, which we submitted into Nature, but it would have been published in January. But we withdrew it because Jim Watson wanted, he said he was close. In any way, he held us up till May. By that time, Jacob and Monod had put the result in their review, you see. So the concept of messenger was there. But basically, it was again an exciting thing, you see. And having all of this, the mRNA, the triplet nature, the genetic code,
Fabulous.
all going on. You know, you would sort of run a sucrose gradient, then go and do some crosses. So it was very exciting. And then, of course, in 1961, the announcement of the Nuremberg experiments. And as Francis was at that conference in Russia, said, well, now, you can determine the code. And we realized that our grand program in molecular genetics, a word which I invented, by the way, which we called our group, the molecular genetics. I mean, it was a great time. All you did is add the word molecular to anything, and you could make yourself unique, you see, just by that way. That we realized that program would not be solved in this manner. And it's about that time that, one began to get a bit restless.
You mentioned something that I want to go back to. You said that when you were in your rooms in Cambridge, and you had this idea that it was this unstable messenger RNA might be the answer. You made the comment that you started talking and Francis understood what you were saying.
Yeah.
This strikes me as what I always imagined, although I never saw happened, when you sat in your shared office in Cambridge and just talked to each other all day long. He was probably one of the few people who understood you at that time.
Yes.
And you must have fed off each other continuously.
Because you see, one's thoughts are, the only way I can describe it is one's thoughts are like balls bouncing up and down, you see. And suddenly you can see two balls bouncing together. But of course, in all the discussions, there are all these added things. So it is the communication of that idea which then clarifies everything for everybody that I think was very good. Now, we never sort of did this. You see, there are people who like to go away, work in a room, work out the entire theory, and then deliver this. I've never been that. I like to sort of have an idea, talk to people, refine it, because sometimes in trying to explain it, I see fatal defects. And everybody who knows me knows that when I start to wander, I found something wrong with this idea, and I better go back and think some more. Or, as sometimes happens, people put an input into it. But I think that whole period was, to me, amazing. We could sit down at coffee and we could discuss it. But the interesting thing is, you could have an idea, go to the thing, and you could do this. Now, Anand Sarabhai was someone who we were discussing, could you initiate proteins? You know, could you get mutants of initiation? So what we would find is that there'd be something interesting if we took a base analog mutant and an acridine mutant. By all theories, they shouldn't suppress each other. So Anand says he'll try this experiment. He went to the cold room, he took the first 2 bottles, put them together, they worked, okay. And I then calculated back, what is the probability that you'd have found those 2 bottles? It was 10 to the minus 5, so about 300 bottles of each. That's why we called it the luck. So this was a time, you know, in which I don't think there's been any more glorious part of doing science than actually, you know, go and have a cup of coffee. And of course, what you could do is if you use pre-warmed plates and you're willing to look with a magnifying glass, you could get the result not overnight, but in about 5 or 6 hours. And so this was wonderful that you could actually prove or disprove a theory within a few hours of formulating it. You know, you didn't have to think of spending years generating everything.
Was this your most exciting phase?
I think this was an exciting phase. I think I've had some other things
You've had many exciting phases.
which are this, but this was amazing because I think it you know, it showed the power of genetic analysis. The next phase was pretty, you know, was a very strategic phase in a sense that Francis and I then started a series of discussions, what we're going to do next, you see. And I think, you know, as I've written in the introduction to the C. elegans about that, we decided that all these other problems are going to be trivial, like the structure of the ribosome, which we might have done, animal viruses, which Jim Watson said we should work on. And so my idea was to take this powerful thing and bring it up to other biological phenomena, not just to proteins and so on.
Harking back to your past.
So I, you know, to go to real biology. And one of the things I've always been interested in is how do nervous systems work? And so I went out to look for this organism which would satisfy all these criteria. I had a few distractions on the way, like getting people to do work on tRNA mutants, which we did, which was very good. That was the origins of doing this. And we needed a system where we had genetics and we had a means of analyzing the mutants. And the analysis was that we are going to be a long way before we can do molecular analysis. We will try on certain things, but for the nervous system, the main thing was, could you get the wiring diagram? Was it, if you had isogenic animals, was it the same?
Yeah.
You know, in all animals. I mean, just to ask elementary questions like that became the ambition of this. And we spent a lot of time exploring the technology of cutting. We knew we had to do electron microscope sections because that's the only way you can resolve whether 2 cells are in contact or not. And therefore we wanted something that fitted into the window. So it had to be a small animal, it had to be easy to grow, it had to have appropriate genetics. And that is how the C. elegans started. And it started simply by trying to get all the pieces of technology to work.
So had you seen C. elegans in a textbook before? Or you
No, I read a remarkable book, I've still got it. It's a product of the University of California Press. It's the micro metazoa. And in this, they had, articles on rotifers. And since I think rotifers are the most sexist animals going, they've reduced the male to just something that makes sperm and just die. The poor male doesn't even have a digestive system. I said, no, I want a real animal with muscles and nerves, not one of these things. In fact, I remember at that time Dictyostelium had been proposed as the organism of the future. And I said, no, I divide organisms into vertebrates, invertebrates and pervertebrates, and Dictyostelium is a member of the pervertebrates. It actually is. All it does is just a kind of more flamboyant sex than any other of these beasts. So, that got started. I isolated, this was great fun, I isolated lots of nematodes from soil. I had a vast collection, in fact. I think you will see it went up to N62. And we looked at all of these to see, and finally, although we thought we'd look at Briggs' eye, we finally resorted to elegans because we got satisfactory microscope sections of it.
So you tried all the nematodes, most of the nematodes?
We tried cutting them all. And of course, I was very fortunate because I wrote a short note and I got some more space. The MRC agreed they would build us an extension. And we would, so one floor would be my extension, which was going to be a new division, but Francis and I kept the two together, as you know. And then the other floor could be an extension for the Sanger group, and the structural studies people needed more space. And the bottom floor was kept empty. It was just a shell. But we covered the floor with paper, and we put a computer in there. And in this place, we spent hours. I mean, we just got locked up with computers there. And it was really pretty good. I mean, I learned a lot about things then. And the whole idea was we're going to use computers to do the serial sections to process it. There was no way we could do it.
Sydney, what was the reaction of the scientific community to your initial emphasis on C. elegans?
Well, I think that everybody thought that it was a kind of joke organism. And nobody thought that any good would come of it. I mean, Jim Watson said to me, "You're 20 years ahead of the time." So I said, "Jim, unless someone gets 20 years ahead of the time, we'll all stay 20 years behind the time." And I think it was a really long-shot project, in the sense it was going to test one important thing. Seymour Benzer also started at the same time, but he thought he could map a relationship between genes and behavior. And he had then started this with Drosophila. But I thought we had to have a different concept of this, namely the genes were used to build a nervous system, which then behaved. In other words, the crossover was what was the nature of the machine. Now, of course, there was a lot of basic work to be done in here, but fortunately, we were able in the MRC to commit to a long-term program like this, to try to bring molecular biology into this. And of course, since structure, but of course in real space, not in Fourier space, is about the same. And as Francis always said, when you can't study a function, do the next best thing, study structure. The idea of asking the following question, which is effectively reformulating what every real geneticist has asked. In other words, he's asking, can I compute phenotypic properties from what I loosely call a wiring diagram? You know, can I get the wiring diagram of a nervous system? And then can I say how it works? Yeah. All right? I mean, I've now realized that this is in total opposition to what is called systems biology, because you see, systems biology tries to solve the inverse problem. What they are saying is, if there's someone playing a drum in a room next door, we're going to record all the sounds, and from the nature of the sounds, we're going to reconstruct the physical properties of that drum. And you can't do it because there's interference and you can't measure it accurately. You can't get a unique solution. Whereas all experimental science has sought to solve forward problems. Okay? Because the argument is get in, get hold of the drum, okay, get its structure, and play it. Yeah. Because once you can do this, you can compute what the sounds would do. So the idea of this science, of this approach to complex systems, is to say, let's get a wiring diagram and let us see whether it is sufficient and necessary to account for the behavior. But what we knew is, we knew we had to master a lot of techniques, and we'd have to just master things, and a lot is owed to people like John. John White came to me as, he was a technician, he had a degree in electronic engineering, and this gave him an opportunity. He got a PhD, but he understood about computers, and as an engineer, he's very inventive. Nicol Thompson came to me from someone else. John Salston was an organic chemist who came there. And we had the people, and they all participated in this. And the important thing was, which I think became clear, what was beautiful about C. elegans, you could have your own problem. Yeah. You could pick it up, and in 3 weeks you could be doing real biology. Yeah. You could get your mutants, and everything was done. Our first paper was published in 1974, but we were only starting to take people into the projects, you know, several years later. And I think that that showed how you could do real genetics. And I think it really sorted out how you do hermaphrodite genetics, the whole business about males and so on. And I think what has been good is that when it migrated to America, then I think that sort of spirit was continued and has been continued ever since. You know, people share their results, people share their information. You'll be amused to know that I think now's the time to go and solve the problem I thought I might solve, which is, can we compute behavior from the wiring diagram? And so I will be starting a small project to see if this is possible.
You're going to do this now?
Yeah, I'm going to do this, yeah. I think I can do this because there are now a lot of people interested in this, and we have a lot of data on C. elegans, and I think it would be good to work out the systems that we need in order to explore problems like this. And I think that, you know, people often ask me, well, what are you working on now? I just say the same problem. The same problem is there with us always. How does the genotype map on the phenotype? You know, how can you take this description of an animal and convert it into an animal? You know, could you actually, as I was saying the other day, could I actually be given a genome sequence and say, my God, that's a zebra. I can see how the stripes appear. Of course, we are so distant from that. Yeah. But I think nobody can be in genetics without doing that. Now, of course, what I now find, and what I think is very important, is in order to do this properly, we have to now really realize we're working with evolutionary constrained animals.
Right.
Okay? We're not working with a designed system. You see, the man in the street doesn't believe in evolution, and you know why? We're trying to tell him the following. You can take a black and white television set, and by random changes, you can turn it into color TV. And he doesn't believe that because he knows if you try and tamper with a television set, you'll break it. So what is the resistance to breakage that we have, you know, in the organisms? What is their structure that you can make changes, but you don't break them all the time? So it tells us that this has got to be organized in a very different way from what we've often considered as partly designed system. The thing I'm most interested in now is the cost of specificity.
The cost of specificity.
Yeah, you see, because there's an evolutionary cost in making everything accurate. So lots of people are out there measuring.
Is everything accurate?
Correct, correct. This is the answer. I think there are lots of don't care conditions. And this means you can't interpret anything about the total behavior if there are things that doesn't matter, if there's 10 times as much stuff as there is. And until you realize this, that is why I think we have to compute it from the properties of the machinery. And why is genetics important? And I think I should say that I was an early supporter of sequencing genomes. But I think the sequencing, allowing you direct contact with the genetic material, allows you to assert there is something there. Yeah. And we can now count up the number of genes and so on. So I think that sequencing will open up the genetics of everything. It's a different style of genetics. Let me tell you, the time I realized this is when Fred Sanger showed me the sequence of lambda. And when I saw that, and I could see all these pieces there, I said, well, you know, this is going to be the answer. Well, people often ask me, you know, what's the next thing to do? What do we do? Well, I think we want to know, which no one seems to be very interested in. I want to answer a very simple question in an organism, which I can answer for C. elegans, but I can't answer it for much more than that, which is, how many different cell types are there? In other words, how many characteristic, non-contingent, that is not response, non-adaptive, gene programs are there? Huh? So I think our big task is actually, it's a little bit to solve that, answer that question, and answer also the question of how they got there. There is an intermediate thing, which are all the molecular assemblages that actually carry out the function. And until we've defined those and know how mutation affects those, we're just going to have a catalog. So I think that genetics has an enormous future. I mean, I think it is the really intellectual challenging part of biology to try to understand this function in terms of this linear description sitting inside you. But the real problem is to understand that, and then hopefully to understand how one kind of organism can evolve into another. What changes are there that do this? So that is why I got very interested in the puffer fish. It was my last model organism. I said I'd always worked on an organism that ate the organism before, you see. So I worked on, if I started with bacteria, well, phage ate E. coli from the inside. But of course, worms eat E. coli from the outside. And of course, I moved to fish because fish eat worms. And the last one, I suppose, will be humans because they eat fish. So I feel that it was quite interesting how exactly that choice was made. I was at Woods Hole in the late 60s and loved to go to the library to read journals. I mean, this browsing has disappeared. And I was reading a journal called The American Naturalist, which I think most people think is a nudie magazine. But effectively, I found these papers there in which the DNA content of several hundred species of fish had been studied. And I was amazed to find that there were fish with 1/8 the amount of DNA. And I used to go to people and say, well, what do you think about this? You know, and people roughly said, well, I'm 8 times better looking than a fish. I'm 8 times smarter. The fish has got the DNA it deserves, which is, of course, it's a joke way of saying that basically the fish is a vertebrate like us with the same body plan, with the same physiological system.
Just needs less DNA.
It just has less DNA.
Yeah.
It's got what I call the discount genome, a discount for sequencing. And I actually tried to get people interested in it.
But how does this fit in with your quest for genetics and function?
Well, you see, this is the thing where I realized I can now do something called comparative genetics, not comparative genomics.
Genetics.
Comparative genetics. I could take a piece of DNA out of the fish and I could say to the mouse, can you read this?
Yes.
Okay, can you read it correctly? And of course we started to do this years ago by looking at control elements in this way, and to our amazement, many of the things are completely conserved. But I want to emphasize it's like a genetic cross, okay? I'm asking the following: can I make a mouse which is exactly the same as the mouse, but instead it's got the control by the fish? I don't want the protein to be the same because that's often very different. But I want to know whether I can use the control. If you look at it, this is no different from making a transductant in the old days. And because I think this technology of recombinant DNA now gives us the opportunity to make these crosses, and I think we can do tremendously interesting things this way. When I look back on how last 60 years or whatever it has been involved very deeply in science, and ask myself, you know, what did I bring to this?
You brought a
well,
lot.
first of all, my motivation was always to solve a puzzle.
Yeah.
I think when I found the solution for it, go on to the next puzzle. And in fact, I've often said that had Francis not locked me up in a room and made me write papers, I wouldn't bother to write the papers. But I think that in looking back, I think what I'm really good at is starting things. See, J.D. Burnell was the one who compared science to a chess game. There's the opening game. There's the mid-game, which most people are playing, the really boring part of science, you know, where you're sort of shuffling pawns around. There's the end game, which very few of us get to play, very few. So rather than stick in the middle game, I like the opening game. I like to say, well, what would it take to get this going, to get a lot of people doing this? I think great labs are distinguished by the people they produce. And I am extremely proud of all the people that have come out to my lab, and when I see them at meetings, and I think, well, you know, it's really fantastic. And when we went to that C. elegans meeting, and there were more than 1,000 people,
1,600.
bright young people, all giving papers, you know, with precision. I mean, top-class science, high-quality science. I think that it's terrific to have started that. And, you know, and hopefully if one could do it more than once, then that's a great legacy to leave. I think today the kind of motivations that are present in science are much more mundane than just the pure intellectual motives. I think, you know, everybody wants to be famous, it's very competitive. People asked me, what does it feel like to win the Nobel Prize? I said, well, you know, it didn't change me at all. I mean, I think I'm still the same person. I said to someone who asked me, what are your long-term aims? And I said, well, for me, long-term and short-term is the same thing. So we just go on to the next thing.
I want to go back and discuss the politics of science.
I like to say over and over again to everybody in politics, there's no applied science, there's no pure science. Science is not mission-oriented. Science, you cannot think of it as basic or fundamental. Science is the best way we know of solving a problem and of finding an answer to things, right? Now we know, in all of our history, we know magic doesn't work. We know religion is unreliable, but science works. And I think that until people come to appreciate this, the fact that we have now a body of experience and of generating knowledge about the world and of solving problems and finding answers to them, and that's all been this activity, you know, that maybe started in the 16th century, and we've just had this ongoing thing. And unless society realizes this, I think this is going to be very difficult. And so, you know, I hope I can go on doing science. So I think there are lots of things to discover. I really like to know why the octopus is so clever.
In thinking about what I wanted to say to you at the end of this, I often return to my friend Jane Hirschfield, the poet, who writes the most beautiful poetry. And I've read through her poems thinking about you and what I wanted to say to you. And there's a poem that I want to read to you that she wrote. And this poem is called Optimism, because you embody all this to me. It goes like this. More and more I have come to admire resilience. Not the simple resistance of a pillow, whose foam returns over and over to the same shape, but the sinuous tenacity of a tree. Finding the light newly blocked on one side, it turns in another. A blind intelligence, true. But out of such persistence arose turtles, rivers, mitochondria, figs. All this resinous, unretractable earth. And to me, Sydney, you are the earth. Thank you very much.
Thank you.